Let's dive right into this incredibly fascinating microscopic journey of how a simple electrical signal from your brain actually becomes physical movement in your muscles. If you're gearing up for your FSC second year or Cambridge A-level biology exams, you already know the sliding filament theory is heavily tested. But today, we're not just going to rotely memorize facts. We're going to visually map out exactly how an invisible electrical impulse triggers a massive coordinated mechanical pull.
Here's the roadmap for our explainer today. We'll quickly cover the anatomy of a muscle, the neuromuscular junction, excitation-contraction coupling, unlocking the muscle, the cross-bridge cycle, and wrap it all up with a rapid-fire exam revision summary.
All right, Section 1, the anatomy of a muscle, building the foundation. Let's get the absolute basics of muscle architecture down so we can anchor the rest of the processes we'll be talking about. You really can't understand the movement if you don't know the parts, right?
Okay, the absolute most crucial starting point is understanding the sarcomere. This is a highly tested definition you simply must have memorized as the fundamental unit of contraction. A muscle fiber is packed with myofibrils, and those are divided into these repeating segments called sarcomeres. The boundaries of a single sarcomere are known as Z-discs. So whenever a muscle contracts, what's actually happening is thousands of these sarcomeres pulling their Z-discs closer together all at the exact same time.
When we zoom in, you can see the structural hierarchy. It's basically thick and thin filaments sandwiched between those Z-discs. This is your visual anchor. The entire magic of muscle contraction happens right here, within this alternating grid of proteins.
Board exams absolutely love asking you to differentiate between these two proteins, so keep this straight. Actin is the thin filament. Just think of it as a smooth, twisted rope. Myosin, on the other hand, is the thick filament, and it's covered in these little extensions called globular heads. These heads, they are literally the microscopic motors of the muscle cell.
Moving on to Section 2, the neuromuscular junction, the brain's command. Let's see exactly how the nervous system tells the muscle it's time to get to work.
What's super interesting here is the physical gap, the synaptic cleft, where the electrical signal has to somehow bridge over to the muscle. This whole area is the neuromuscular junction. Up top, the nerve terminal is packed with vesicles containing a chemical messenger, a neurotransmitter called acetylcholine. Down below it is the muscle fiber membrane, known as the motor end plate, just waiting for the signal.
So here's your chronological, high-yield sequence of events at the neuromuscular junction. You're going to want to memorize these 5 steps perfectly for your short answer questions. First, the electrical action potential hits the nerve terminal. This forces voltage-gated calcium channels to open. Calcium rushes in, and that forces the vesicles to dump acetylcholine straight into the gap. The ACH then crosses over and binds to nicotinic receptors on the muscle. Think of this like flipping a switch. It opens sodium channels on the muscle, causing a massive influx of sodium. That sudden electrical shift on the muscle is your end plate potential.
Which brings us to Section 3, excitation-contraction coupling, the internal trigger. Once that signal crosses the gap, the electrical spark dives deep into the muscle fiber. Those electrical signals travel down these specialized channels called T-tubules. They dive deep inside, right up against a webbing called the Sarcoplasmic reticulum. The SR is basically a giant storage locker for calcium. So when the electricity hits, that locker bursts wide open, completely flooding the muscle cell with calcium ions.
I want to pause for a second here and ask a question that represents one of the biggest exam traps students fall into. When a muscle visibly bulges and shortens, do the individual actin and myosin filaments physically shrink? Do they get cut down in size?
Please absolutely sear this into your memory. No, the actin and myosin filaments do not shrink. They only slide past each other. Never write that the filaments change length on your exam. The sarcomere shortens because the filaments pull themselves past one another, overlapping more deeply, but the actual proteins themselves stay the exact same size. All right, Section 4, unlocking the muscle, the calcium key. Let's unpack how that huge flood of calcium actually prepares the filaments for that sliding action. Think of this as a classic lock and key mechanism, where you must differentiate these two perfectly. Troponin is the lock, and tropomyosin is the physical door blocking the way. Let's use an analogy here. Tropomyosin is a long protein that winds all the way around the actin filament, completely covering up the specific spots where myosin actually wants to attach. It's a locked door. Troponin is the complex sitting right on top of that door, acting as the padlock.
So, when the calcium key binds to the troponin lock, it swings that tropomyosin door wide open, exposing the actin binding sites so the muscle can finally contract. That calcium that just flooded out of the sarcoplasmic reticulum acts as the key. It binds directly to the troponin. This binding causes troponin to totally change its shape, literally dragging the tropomyosin ribbon away from the binding sites on the actin. And boom, suddenly the myosin heads have a clear target.
Moving into Section 5, the cross-bridge cycle, the power stroke. The lock is open, so now it's time to pull.
This cycle brilliantly illustrates the continuous circular nature of the sliding filament model that we need to break down step by step. The whole process requires two main things: the exposed binding sites on actin, which we literally just achieved using calcium, and energy in the form of ATP. Think of the myosin head like an oar rowing a boat. It binds, pulls the actin toward the center during the power stroke, and then requires fresh ATP just to let go and recock. Walk through this cycle with me. First, the energized myosin head grabs the exposed actin. That's your cross bridge. Next, it releases an inorganic phosphate, causing the head to pivot and bend. This is the power stroke, essentially pulling the actin filament toward the center. Now here's the crazy part. The myosin is stuck. It will absolutely not let go of the actin until a fresh molecule of ATP binds to it. Once ATP binds, the head finally detaches. Then that ATP is broken down into ADP, which releases enough energy to recock the head back to its starting position, totally ready to grab again. The really crucial takeaway here is combining the micro and macro views. The microscopic rowing of the cross bridges is what physically pulls those Z-discs closer together on the macroscopic level. You have full sarcomere shrinking as the bands overlap because those exact molecular engines, the cross bridges, are ratcheting forward to make that happen. And this cycle repeats hundreds of times per second as long as calcium and ATP are around.
For our high-yield exam corner, you've got to know that without ATP to detach the myosin, the cross bridges stay permanently locked. This perfectly explains the stiffening of a body known as rigor mortis. It's kind of macabre, but it's an incredible biological example. After breathing stops, ATP production stops. And since, as we just learned, ATP is strictly required to break the cross bridge and relax the muscle, the myosin heads basically just become stuck to the actin. The muscles literally lock in place until the proteins eventually start to degrade.
Okay, final stretch. Section 6, Exam Revision Summary, the 10-second takeaway. Let's bring everything we've talked about together so you are 100% prepared for exam day.
This right here is your absolute must-know 10-second chronological checklist for the entire muscle contraction process. Signal arrives, acetylcholine crosses the gap. The electrical wave opens the calcium lockers. Calcium unlocks the troponin tropomyosin door. Myosin reaches up, forms a cross bridge, and pulls. And finally, ATP comes in to break the bridge and reset the whole system. If you can write out these 5 basic steps, you're going to absolutely crush any long answer question on this topic.
I want to leave you with this final thought to ponder. If ATP is the energy needed to relax the muscle, what exactly is happening internally when you experience a sudden, super painful muscle cramp? Think about your body's energy demands during a long run, oxygen depletion, and how the absence of that crucial ATP might just leave your internal machinery completely stuck mid-row. It totally changes how you think about a simple Charlie horse, right? Thanks for walking through this incredible biological machinery with me. Keep utilizing this explainer for your revision, and best of luck with your studies.